Hello students, I am Dr. Suman from Victree classes. Today I am going to teach you about Chemiosmotic hypothesis, very important topic in photosynthesis chapter given by P. Mitchell, right? And before knowing this, you should know the plant anatomy. Let us study structure of chloroplast. So in this diagram you can see chloroplast has two membranes. One is the outer membrane, next is the inner membrane. Then you can see there are disc-shaped structures placed one above the other. This one structure is called thylakoid. Then if they are stacked one above the another, they are called a granum. Stack of thylakoids is called a granum. The space base is called the stroma. Then the space inside the thylakoid, that is called the lumen. I have marked it, you can see, right? Now after this, let us study the thylakoid membrane as such, because all the reactions are going to take place in and around the thylakoid membrane. Now the space outside the thylakoid membrane, that is called the stroma. Now the space inside the thylakoid membrane is called the lumen. Now next you can see there are various photosystems. First you have P680, that is photosystem 2. That is a site for splitting of water molecule. Then you have a mobile carrier plastoquinone. After that you have a complex of cytochrome B6 and F. Along with it you have another mobile carrier called plastocyanin, right? Then you have photosystem 1, that is P700, right? Now as you can see on the inner side of photosystem 2, that is inner side of thylakoid membrane, that is a site for splitting a water molecule. You should remember splitting a water molecule takes place on the inside of the thylakoid membrane, right? Now it will give various products like H plus, O2 and 1 electron. After splitting a molecule, now you can see in the diagram along with PS2 there is another important structure called ferredoxin, right? So this is attached to P700. Now on the lower side you can see there is a complex of F0 and F1 called ATPase complex. This is a site from where the ATP will be synthesized. I am writing the names now. PQ means plastoquinone, PC is plastocyanin, and FD means ferredoxin, right? And FNR, that is an enzyme which will be needed, that is called ferredoxin NADPH reductase enzyme. This plays an important role for the production of NADPH, right? Okay. Now in the next, let us study the process. Now after understanding the location of all the systems, let us see the process. As light falls on PS2, it gets activated and releases one electron. This electron transfers to the plastoquinone. Now this plastoquinone will pass the electron to cytochrome B6F complex, but along with it, it will allow the entry of one H plus ion from the stroma to the lumen. So you have to remember that plastoquinone will allow the entry of H plus ion along with the electron. Now once the electron is transferred to cytochrome B6F, it will further get transferred to plastocyanin. From the plastocyanin, it will go to PS1, right? Here, now in PS1, when the electron reaches here, it will go to ferredoxin. Now here, ferredoxin in the presence of FNR enzyme, it will convert NADP into NADPH in the presence of H plus ion plus 1 electron, right? So ferredoxin in the presence of FNR enzyme converts NADP with the help of 1 electron and H plus from the stroma into NADPH plus 1 H plus ion or 1 proton, right? Now on the inside of the thylakoid membrane, as I told you, there is splitting a water molecule. So water molecule splits to form 1 electron one proton and oxygen gas. This electron will go to PS2 and compensate for the loss of electron, right? Similarly, the cyanine that will transfer the electron to P700 or PS1 and compensate for the loss of electron which has been transferred to ferredoxide, right? So that the electron remains with PS1 and PS2. Now when the H plus at concentration increases due to splitting of water molecule, in the lumen you can see there are so many protons, they are increasing in number. Now here the concentration of H plus increases, and in the stroma, as all the H plus is passing through plastoquinone, the concentration of H plus ion will decrease in the stroma, right? This will provide energy for CF0, and this in the presence of ATPase enzyme will activate CF1 and convert ADP along with inorganic phosphate into ATP. So you are getting two end products, that is NADPH and ATP in this whole process, right? Now the whole process is taking place due to the proton gradient, difference in proton gradient between the stroma and the lumen. When the concentration of protons increases, that activates the CF0 and CF1 to convert ADP to ATP. Let us study the theory now. Given by Peter Mitchell in 1961, here in this process electron transport produces a proton gradient. This develops on the inner side of the thylakoid membrane. As the lumen is enriched with proton ions, one molecule ATP is formed when 3 protons pass through ATP synthase. So this is from my side today, and see you later with other topics. Thank you.